Hey everyone, this is Immune System video number 2, and in this video we are going to cover innate immunity, the second line of defense, and these are internal non-specific defenses. If a pathogen gets past the first line of defense, skin and mucosal membranes, what is going to do battle internally? And so the second line of defense is made up of a number of cells, some chemicals, specific type of proteins that we'll be talking about. There are a couple of responses involved, the inflammatory and response and fever. But we're going to take up those number 3 and number 4 in a later video. For this one, number 1 and number 2. All right, and so this slide is just showing first line of defense over here, skin and mucous membranes and secretions from those. And what if something gets by, that's first line of defense. So it's most likely if you have a cut in your skin, or there are some pathogens that are able to, get by the first line of defense. Then the second line of defense, non-specific internal defenses come into play. These are the two main processes, fever and inflammation, we'll talk about later. These are the topics for this video. What are the non-specific cells that are going to mount to defense? So there are a number of different types, and they're kind of grouped into those that have similar action. So one type are the neutrophils, the macrophages, and the dendritic cells. They're all types of phagocytic cells. Another type are the basophils and the mast cells. These are cells that are armed with a set of chemicals called pro-inflammatory chemicals. And the natural killer cells are targeted cells that are targeted to abnormal body cells. And finally, eosinophils are very effective against parasitic infections. So those are a group of cells that are involved in non-specific defense. On the other side are shown proteins that are involved. We're going to talk about two main types. And these are classes of proteins. So there's a number of proteins in each of these two groups. One are the interferons. Interferons are especially effective against viral infections. And we'll talk about how that comes about. Another set of proteins are the complement proteins. These are circulating in the bloodstream, and these are particularly effective against bacterial infections. And they trigger a number of immune defenses to kind of pile on, to cooperate in this defense against internal bacterial infection. All right, so let's go down the list, starting with the cells. First on the list for cells are neutrophils, macrophages, and dendritic cells. So what's common between all of these is that they are types of phagocytic cells. If they encounter a pathogen, they can engulf it, internalize it, and break it down. There are some subtle differences between them. So neutrophils, for example, these are common in the bloodstream. They're circulating. But if there's some kind of infection or injury, there's an alarm which attracts neutrophils to the site. And so they exit from the bloodstream. They are usually the first cells to arrive on some kind of infection due to the inflammatory response. Macrophages are not circulating cells. These are cells are present in the tissues. Most body tissues have some kind of macrophage that's resident in the tissues. These cells are able to move around. They're motile cells, so they can squeeze between the cells in a tissue. And they will arrive on a site of damage or infection, not as quickly as the neutrophils, but they tend to hang around longer during the battle and the cleanup process that results. Third type of phagocytic cell are the dendritic cells. So the dendritic cells are kind of unique in that when they internalize a pathogen, they take a bit of that internalized pathogen and they put it on their surface. And this antigen presentation, it's called, is involved in activating cells of the adaptive immune system, the lymphocytes, T and B cells. So they're kind of bridging between the innate immunity and the adaptive immunity. All right, just a few slides here. This slide is illustrating a macrophage and a dendritic cell. Macrophages are macro for large cells, phage for phagocytic. So here's a pictorial just demonstrating a macrophage internalizing a bacterium and degrading it and then releasing the degraded contents. Dendritic cells are named dendritic cells because they have these multiple plasma membrane extensions called dendrites. When they internalize a pathogen and break it down, they stick it on the surface like this, and that's the signal for cells of the adaptive immune system to latch on and activate. I have a short video showing a macrophage in action, how it's motile and how it's also internalizing a bacterium. Let's take a look. So there's the macrophage traveling around, and the little black dots are bacteria. And this is in a cell culture plate, but a similar type of thing would be happening inside the body. Eventually, the macrophage catches up, latches on, and internalizes the bacterium. And this video doesn't go to the point where it's degraded, but that would be happening next. All right, in addition to the macrophages, neutrophils, and dendritic cells, another type of non-specific internal cell defense are the basophils and the mast cells. So these cells are very similar in terms of what they contain and what they can trigger. The main difference is basophils are the circulating version. They're present in the bloodstream. And mast cells are the non-circulating version. They're present in the tissues. Basophils can leave the circulation and occupy the tissues, and then they're called mast cells, basically. So both of these cell types are chock full of a set of chemicals that are called pro-inflammatory chemicals. And when a mast cell gets triggered, and this would happen because of an infection or damage to a tissue, they will release all these chemicals. And this is like a chemical alarm, basically, which triggers the inflammatory response. We'll talk about the inflammatory response in the next video, but what are the chemicals? So histamine is a type of inflammatory chemical, and this is going to vasodilate local blood vessels. And it's going to increase permeability of capillaries in the location. Also, heparin gets released. This acts to prevent coagulation at a site. The net effect of this is more blood is going to be delivered to a site of infection or damage, and it's going to deliver necessary chemicals to help battle or repair what's going on. They also release another type of pro-inflammatory chemical, eicosanoids, and these are going to serve to increase inflammation, and they're going to attract other immune system cells to the site. So basically what's happening here for a basophil or a mast cell, these are the soup that they released when triggered by damage or infection. These have effects on the circulation, which is going to allow for more materials to arrive, to do battle, or to repair any kind of tissue damage. So that's the basic idea of the inflammatory response. And these cells, the mast cells and the basophils, are the cells that contain the molecules that trigger that response. Pro-inflammatory chemicals. Okay, another type of non-specific cell are the natural killer cells. You could probably guess what these cells do, but what they specifically are targeted against are abnormal body cells. And two really good examples of that are cells that are virus or bacteria infected, mostly virus infected cells. viruses require entry into a cell in order to reproduce. So when they enter a cell, they can sort of hide from the immune system, except for the fact that they cause cells that they're inhabiting to make chemical proteins that they put on their surface. Natural killer cells are able to detect those. Okay? Tumor cells are another example. Tumor cells put abnormal proteins on their surface, and that betrays them as an abnormal cell. Natural killer cells are usually circulating in the blood or as part of a lymphatic nodule, remember, a group of immune system cells in a secondary lymphatic structure, such as a lymph node or the spleen. Now, the way they kill abnormal cells is they release a 1-2 punch of chemicals that include one type of protein called perforin. Perforin punctures holes in the abnormal cell. And then the second punch, I guess, is a type of enzyme called granzymes. These granzymes enter through the hole produced by perforin, and they start degrading the abnormal cell from the inside. Eventually, the cell shrivels up and dies, and that's called apoptosis. So natural killer cells patrol the body looking for abnormal cells. When they encounter them, they puncture them, causing lysis of the cell, and then they kill them by releasing enzymes that degrade them from the inside. And this is shown on this slide. Here's a natural killer cell. Let's say it's a virus-infected cell. Natural killer cell recognizes it as abnormal, releases these perforins, the blue spikes here. That creates a pore of entry for the granzymes, the little pale balls are the enzymes that enter this abnormal cell, break it down, and it shrivels up and undergoes cell death. Last type of cells are the eosinophils. Eosinophils are cells that are packed full of degradatives and enzymes in vesicles, similar to lysosomes. So these are very potent. And the feature of eosinophils that makes them unique is that they're able to release the contents of these vesicles to the outside. So they're very good at targeting parasitic infections. And what they do is eosinophils will line up on the surface of a parasite, release their degradative enzymes onto the surface, and basically cause the parasite to be degraded from the outside. And that's shown here on this little mini movie here. Here's a parasitic worm. Here are eosinophils basically ganging up, latching onto it on the outside. They're going to release the contents of their granules, which are degradative enzymes, which are going to start degrading the worm from the outside. All right, so all of these cells are nonspecific cells in the sense that they're not targeting a specific pathogen. Eosinophils target parasitic worms, but it's not like it's targeted to a specific type of worm. It's just that they're effective against that type of pathogen. Okay? So those are the cells that make up the second line of defense, the nonspecific cellular internal defenses. Let's talk about the proteins that are involved in this as well. Okay, two types of proteins. One is a class of proteins called interferons. Interferons are especially effective against viral pathogens. And what interferons do is they get released from a cell that's already infected with a virus. Like I said, viruses reproduce. They can't reproduce by themselves. They have to enter into a cell and use the cellular machinery to make new virus particles. When a virus enters a cell, one of the first things a cell does is release interferons. These interferons are going to bump into nearby cells to the one that was infected. So some of these cells will just be neighboring cells in the tissue, same type as the infected cell. In the neighboring cells, interferon triggers them, alerts them basically to start making chemicals that are going to prevent the virus from infecting them or reproducing inside of them. Okay, so interferons cause neighboring cells to interfere with virus infection. All right, that's one thing they'll do. Sound the alarm to neighboring cells. The other thing interferons do is they stimulate other nearby cells of the innate immunity, the macrophages and natural killer cells that we just mentioned. So they're going to stimulate these cells to be attracted to the site of this virus-infected cell, and they're going to cause them to do their thing, which is shown on this slide. All right, so suppose this cell here gets infected by a virus, and the first thing that an infected cell will be doing is to releasing interferons. So IFN stands for interferon. There are different types of interferon that are indicated by alpha, beta, gamma here. But what they're going to do is act on a nearby neighboring cell, cause this cell to make enzymes and other proteins that are going to prevent this cell from being infected by the same virus. Okay, so this is alerted neighboring cells in the tissue. Interferons also cause natural killer cells to become more active. So this natural killer cell is going to be attracted now to this abnormal virus-infected cell. And when it encounters it, it's going to release perforins and granzymes, causing the cell to die. Not only that, interferons are going to alert macrophages, which are going to be attracted to this abnormal cell to latch onto it and take it in by phagocytosis to break it down. So interferons prevent spread of the virus in normal cells, and they attract other cells to kill the virus-infected cell. The other example of nonspecific immune defenses through proteins are a set of proteins called the complement proteins. The complement proteins are a set of about 30 or so proteins that are circulating in the blood plasma. These are made by the liver, and they're continuously released in inactive form. So what you normally have circulating is all of these complement proteins that are inactive. They can be switched to active form by an infection. If you have a bacterial infection, that's the trigger that will switch. Okay? And when they're switched to active form, then you have this set of complement proteins now that are going to carry out a bunch of actions to help to battle a bacterial infection. So what are those actions? So they're listed on this slide and the next slide, and then illustrated on the final slide. One action of complement is to coat the pathogen. If it's a bacterium, the complement proteins latch onto it, basically paint it with these complement proteins. What does that do? That serves as a signal for phagocytes, macrophages, neutrophils, dendritic cells to take up this coated pathogen. Okay, so opsonization coats the pathogen with complement proteins and makes phagocytosis much more likely. Second thing that complement proteins do when they're activated is trigger the inflammatory response. So they are going to alert the mast cells and the basophils to release their storm of inflammatory chemicals. And that's going to send the alarm to the rest of the immune system. Third thing that complement proteins do is they lyse the pathogen. So they assemble into a set of proteins called the MAC complex, membrane attack complex, punch holes in the bacterial cell wall, and lyse the cell. And that's going to kill it. A last thing that they do is they help to clean up by complement proteins latching on to antibodies that have bound to abnormal pathogens. And then what happens is the complement attaches this structure to a red blood cell. And when the red blood cell circulates to the spleen, the macrophages in the spleen get rid of it. They basically break it down. So they're targeted for degradation. And those four actions of the complement proteins are illustrated here. Here's a complement protein latching onto a pathogen, opsonizing it, making it more likely to be phagocytized. Here are complement proteins activating the inflammatory response. And it's going to attract various cells of the innate immune response. Here they are forming an attack complex, lysing this pathogen. And here's complement attaching to an antibody bound to some pathogen and linking it to red blood cells. And that's going to make this destined for degradation in the spleen. All right, so that's a description of part of the innate immune responses, the cells and the main proteins that are involved. In the next video, we'll talk about the inflammatory response. See ya.